Okay, we're recording. So, yeah, hi. So you are
Hello.
Rareș, hi, and I'm Tzvi.
Yes.
Um, and yeah, I guess what we're here to talk about is there's the general idea of iterated meiotic selection, where um, you take a set of input cells which um, have different genomes, and then you want to um, repeatedly do two operations. One of them is inducing meiosis. So you get a
Yes.
diploid cell with two of each chromosome to divide and recombine. So you get crossovers, which produces a bunch of haploid cells that have chromosomes that are combinations of the original two. So that's one operation. You can induce meiosis in a diploid cell line, and then the other operation is fusing two haploid cell lines to get a diploid cell. Um, and then the question is, and so this could in theory be a powerful technique for making a diploid cell or a haploid cell that has a genome that you like for whatever reason. It has low mutation rate, or it scores highly on a polygenic score, like for health or intelligence, blah blah blah. um,
Yes.
and then there's the question that I'm curious about, which is uh, yeah, there's like this math or CS or reinforcement learning problem of what is this if you actually have a set of different cells like in a lab, you have to choose which cells to apply the two operations to. You have to choose which cells do I want to make to induce meiosis in, and and then which cell lines do I want to fuse into
Yes.
two haploids into a diploid. Um, so you sent me some stuff. I think one of the things you were talking about was the sort of the linear idea, where if we are just looking at a single chromosome, then um, we can sort of combine uh, we can combine things so that we get the first segment the way we want it, then the next segment, then the next segment.
Yes. The idea which I said was essentially the simplest, and of course inefficient, but simplest possible way to get a chromosome with maximum score for a single, this is important for a single metric. Of course, if there are multiple metrics we care about and each segment has a different score on both of them, this will not give necessarily a maximum that we like. But for a single possible metric, there is a simple linear algorithm that works. Though, of course, in a real scenario where we wish to parallelize, it won't be as efficient.
So I think I'm not even sure that even if we use the strategy you're outlining, I think there's still questions here because you know so like first of all, um, there's a question. So let me just check that I understand the strategy you're imagining. Are you imagining basically we start off with let's just say we have two cell lines, each of them we're just looking at a single chromosome. So the cell line number one has one chromosome, and cell line number two has the other chromosome, and then
Yes.
we're going to fuse those, and then we're going to induce meiosis, and we're going to induce meiosis until we get the first crossover in the position that we want. Is that right?
Yes.
So I would say
The
even there's already a choice that we have to make because um, I don't know if you see where I'm going. Like basically, you have to choose how like you can that there's some cost to running each every operation. Like if we break down that protocol, you would have to be so once you do fusion, you do fusion and then you induce meiosis. So I assume we have like a cell culture with the diploid cell that's dividing, and then we're going to induce meiosis probably in a big batch, like en masse. So now we have a whole bunch of cells. A lot of them might still be diploid. Some of them will be haploid. And then we're going to try to isolate some cell lines that are haploid. And then we have to verify, we have to sequence them. So we're just going to assume that they can be cultured, that the haploid lines can be cultured. But we don't have to assume that. That's another assumption that you could vary. But if we assume that, you have to pick a number of cultures to grow, of haploid cell line cultures to grow. You see what I'm saying?
Yeah. Yes.
You could do a whole bunch, but that might be expensive. I mean, you have to. It's not very expensive. You have to, whatever procedure you're doing to isolate it, which might be, probably you can use like some flow, like a cell cytometry thing, where you're sorting by mass, I guess, because the haploid cells should be smaller, or some visual markers or something. But facts, but it's still a choice. So in the mathematical model, there's like a choice of like how many cultures are you growing. So there's some balance where there's a cost to how many cultures the more cultures you have, the larger the cost. But on the other hand, if you only do like 3 cultures, then you probably won't get the crossover you want.
Yes, correct. The number of cultures should be larger than the amount of segments to not to to guarantee that there is for each segment a maximum, a proper maximum. Because if we only have 3 and there are, let's say, 10 segments, there's only in each particular spot, in which particular segment, there's only a maximum of from those 3.
Mm-hmm. I mean, I assume that the optimal number just depends on like what the cost function is. Like if you if it's very costly to take a longer amount of time, but not very costly to have more cell cultures that you then sequence. So if you want to go really fast, then you want to have a large number of cell lines. If it's fine to go slow, but you don't want to have a lot of cell lines that you're culturing, then you want to do a smaller number. And then if you fail to get the crossover you want, you just do it again. Oh, are you frozen?
It's okay now.
Okay.
Yes, I understood.
Okay.
In case of the sample simulations you have given, the number of colors we can say are equivalent to the number of unique chromosomes, right? If I understood correctly.
Wait, say it again.
The number of colors each corresponds to unique chromosome with a unique score, unique distribution of its segments, correct?
Wait, why is that the number of cultures? I'm not sure I'm understanding
asking,
Are we still talking about this context where we have just the two? Wait, let me try to set up a some kind of screen share thing. Um, just give me a minute. Um, if I understand what you're asking, wait, are you just asking how many cell lines do we have at all? Because I think we need to have a lot more cell lines than just the number of starting chromosomes. Because the issue is that we need to have a cell line in order to sequence the cell. Does that make sense?
Yes, of course.
Does that answer your question, or?
Yes, I understand. Thank you.
Okay, I'm still not sure I understood the question, but yeah, like we need many cell lines because you need a cell line in order to sequence the cell line. So yeah, I think that the linear strategy doesn't take out quite
Of
efficient.
course, it's inefficient. I just meant to show that there is some baseline of success, even if very inefficient and for a very limited case.
Gotcha. Yeah.
In the simulations, simulation I've given you, I've implemented it, so it can be there.
Um, so yeah, what do you think about the next step is for doing
Well,
RL
the
to the more
most
efficient?
relevant part, of course, is setting up the environment. What's the action space, the state space, and based on that, if the space is well defined well enough, then even very basic learning or whatever solutions would be applied as they are to see if they work and to what extent, because they know they may be inefficient. It's mostly, I think, at least in the early stages before we get into multiple objectives and like how to set the reward, because there are multiple things to be balanced besides the score, the number of crowds to be minimized.
Yeah. Well, we don't necessarily have to minimize the number of crossovers, but I think that, well, you're right that we would. I think we would end up minimizing the
On
number of
one
crossovers
chromosome, say
because
minimize it on one,
if you're introducing
so
more
the
crossovers,
solution can be parallelized because the rounds is what is important, minimizing the number of rounds on the entire group of every chromosome.
mm. Oh yeah, maybe we should just first talk. You may have seen this in the um, in what I had written, but I think that you should. I think probably like for this project, at least at first, it's best to either focus on the chromosome selection aspect or to focus on the single chromosome scenario. Like there's sort of it's not really, you can't really separate things, but okay, sorry, I'm just gonna interrupt myself and try to share my screen. Let's see. Yeah, exactly. Let me, can you see?
Yes, it's loading. I can see it now.
I'm now sharing. Can you see that?
The simulation that I had made was with IS in mind.
It's like a blank Google Draw thing. I think trying to record this on my computer on extremely slowly. Sorry about that. But okay, so um, I would want to say that in order, like, full IMS, full iterated meiotic selection, is like pretty complicated. Or at least, I mean, I don't understand it, is what I mean. And so I would sort of want to analyze it as kind of being two separate processes. One of the processes is, as I said, permanent chromosome selection. In other words, moving chromosomes from cells, and then the other process is the like building up crossed over chromosomes, where you're you're taking chromosomes all of the same number and then trying to build up a chromosome that has good segments from different chromosomes. And so sorry, my computer's so slow right now. So this is the chromosome selection aspect. And then, like, sort of orthogonally or running in parallel to that, you have the crossovers thing. And so I would sort of think of it as, if you're actually doing iterated meiotic selection, you have these two separate goals. First of all, you're trying to make there exist anywhere within your cell population a really good chromosome number 1. And then you're also trying to make there exist somewhere in the cell population a really good chromosome number 2. It might be in a different cell, and so on, for each chromosome number. And then you're trying to, and then sort of separately, you're trying to bring them together, bring the good chromosomes with the good crossovers together into one cell line. And yeah, and so so therefore, and like, there's an extra level of complicatedness where you're trying to do both of those things. And so I would, like, in real life, to actually use the reinforcement learning thing to actually make decisions about scheduling, you of course would want to use the whole the full iterated meiotic selection scenario.
I understand. Oh, sorry, it was a bit unclear. I get it now. The search that I made treated a chromosome in a vacuum, let's say. That's why I disregard this aspect. But yes, I understand better now.
But so, I mean, I guess what I was sort of trying to get at was, it actually does make, I think it does make sense to treat it in a vacuum. Just say, we're going to only look at one chromosome, and then we're going to do reinforcement learning just focusing on this one chromosome, and that will sort of teach us about that scenario. Then, as a totally separate thing, you can do reinforcement learning to the other thing, the poor man's chromosome selection, where you're doing iterated
trying to do it on a larger scale and treating only the scores as the score of each individual chromosome without taking into account their segments.
Yeah, or like so the state model, the world model, in the first half with just doing a single chromosome number, the world model or the state is like you have a set of cells, some of them are diploid, some of them are haploid. A diploid cell has just 2 copies of this chromosome, and the haploid cells have 1 copy of this chromosome, and they have different segments. That's the world state. And then the actions that you're allowed to do are induce meiosis or fuse 2 cells. And you can get much more granular with the action space, and you probably want to.
Yes.
But yeah. And then so that's the state space for the single chromosome one, and then for the state space for the poor man's chromosome selection thing is we don't have any crossover. We just have a cell that has a bunch of chromosomes, like 23 chromosomes, if it's haploid, or 46 chromosomes. And yeah, as you say, we don't have segments. We just have a score for each chromosome, and then we're trying to do, and then you have this like fake meiosis operation that isn't necessarily biologically realistic, but where you can
extract a chromosome from plant it into another, if I remember correctly.
Well, that would sort of avoid this whole thing. If you could do, yeah. I would like to be able to do this. We might be able to do it, but that's like a separate research project. If we assume we only have iterated meiotic selection, there's sort of a separate idea of like, I don't know, it's not very biologically realistic, but just to understand the math, it's like if you have a diploid cell, you could make up this fake operation, which is like meiosis, but with no crossover. Basically, you have a diploid cell, and then you randomly choose a chromosome number 1, randomly choose a chromosome number 2, randomly choose a chromosome number 3. So you get a haploid cell, and it's sort of like a meiotic daughter cell of the diploid cell, but it's just whole chromosomes rather than like crossed over chromosomes.
So sort of cut instead of properly divided cell.
Yeah, like cut in half. Yeah.
Okay.
So that's, I mean, I'm bringing that up just because it's like a somewhat simpler, it's like a fairly simple, I mean, it's not that much simpler, but you know, if someone's working on this project, such as you, say what?
Well, it is simpler. It lacks the crossover operation.
Yeah. It's not like that. It's not like so much simpler, but it's at least somewhat simpler. And it's just like mathematically, this is like one principal way, I think, for us to sort of get more understand. There's at least 2 goals here with like investigating this protocol IMS with reinforcement learning. One of them would be to like actually make a good scheduler, like that you could actually use to run a biological procedure. Another goal is to more conceptually understand what's going on, like conceptually how efficient can it be and why is it efficient or inefficient, and how could you, like, for example, this might inform how to make it more efficient. Like suppose that I could spend 6 months doing research to either make meiosis happen 2 days faster, or I could make it so that the haploid cells divide half a day faster, or I could make it so that the cell fusion is that much more efficient, or whatever. Like there's different things I could try to research to fix, and And if I understood why the thing is fast or slow, then I could know what to research.
Yes, and for this particular issue, I think that if an environment is present and out there, other people can easily attempt better solutions, which might speed up this particular problem
Mm-hmm.
even faster. Because RL, my intuition, even for previously unattempted problems like this, can have really quick development. Because this is even this domain, I think, than the standard RL problems of complex games. So I think this is an area where yes, progress can be quick somewhat relative to others. And I wanted to ask, you said to a scheduler. Is this scheduler strictly prescribing a solution, as in for this particular collection of cells and chromosomes, these are the steps that should be taken, or is it something more a part of a more evolved mechanism that automatically does this separation?
Is it part of a mechanism that...
So is it just a list of actions that can be performed by a researcher or some other means, or is it part of a larger technical solution that is more autonomous?
I want to be agnostic about that. So you're asking are we directly plugging in the outputs of our reinforcement learning agent in some scenario? Are we directly applying that to a robot lab or something, or is it just the researcher looks at that? I think
yes.
I think basically, in real life, the answer probably would be, at least at first, for a while, it would be the researcher. A researcher does it.
Right. I'm asking because that can give it's a lesser cap on inference times and can output multiple solutions. All right, thank you.
Yeah.
So in a sense, there are two particular ways to approach this one, which is a lot more abstract, which does not take into account the poor man's chromosome selection portion of the operation, and one which does for the entire IMS.
Yeah, well, I would say that there's at least three schemes. You could do just a single chromosome separately. You could do just poor man's chromosome selection. And then thirdly, you could do both of them together, where the state is a haploid cell has several different chromosomes of different numbers, and the chromosomes have subsegment scores. And when you make a diploid cell do meiosis, it produces crossover recombined chromosomes for each chromosome number.
I understand.
Yeah. So, I think a big thing here is to be clear. I think the state space, there's a whole lot of choice in the state space, I would say. So first of all, you can have hidden state, where in real life you have hidden state for a few reasons, right? So first of all, um, you don't actually ever know the genome with 100% certainty.
Of
Uh,
course.
you can get pretty close if you have a cell line that, if you have like a monoclone that you then cultured for a long time and got like lots of cells, then you can get pretty close to certainty. Um, but um,
Mm, wouldn't state space with large uncertainty be undesirable for those who might want to make use of these technologies in the future? They might not appreciate, even in theory, the existence of such large uncertainty, especially if in an RL algorithm it has to be baked in.
what do you mean by being baked into the RL?
Uh, Hidden states, the fact that hidden states exist modify them somewhat.
Um,
A partially observed Markov decision process is qualitatively different from a normal one.
mm-hmm. Yeah, well, I mean, I think the reality just is that it has hidden state, so it's partially observed Markov.
Of course, but I'm wondering if at this particular stage, if that uh, should be present already,
Oh, not
or
necessarily. Yeah.
if the assumption of 100% or near 100% certainty is unrealistic.
Uh, well, it's not. I don't know if it's that unrealistic because you can just culture a cell for a while, and then, um, get pretty confident. To put it a different way. So, okay, the simplest thing that one could do that would still be useful would be, or a simpler thing one could do would be, you just assume that the agent always knows the genome or the sequence of every cell line
Yes,
immediately. That's all right. It still can be useful because you can still model the cost of maintaining many cell lines and choosing which ones to do meiosis and which ones to
yes,
pair up. Yeah.
of course.
So that's already useful. So I'm not really. Well, the thing I'm saying right now is not very relevant to right now, except basically the reason I'm saying it is that, so we discussed over email, like it would be cool to have this be like a sort of a somewhat general, like an extensible framework or whatever, where it's not just.
Also, have the uncertainty in the framework already, which can be turned off if desired.
Yeah, or like not necessarily. Or some, I don't. Yeah, I don't have enough experience with this to even know what it would look like. But somehow be thinking when thinking when figuring out the architecture for the RL framework, like when making decisions about how to set up the state space, keeping in mind that there might be quite significant changes we could want to make later, such as adding in hidden state. Um,
Of course, I'm hoping the standard frameworks have this included, but I'll check, of course.
mhm. um, yeah, and then there's just also all sorts of other things that you could put in the model if you want or not. So, for example, when you culture cells, they always have a low rate, a low but non-zero rate of de novo mutation. Um,
Yes,
so that's another thing.
mutations which are assumed to be most of the time deleterious.
Yeah, I think we would like. I think the real thing is most of the time they don't matter, and then occasionally they are deleterious, and then very, very rarely they are beneficial.
Okay.
So one, simple-ish way to model that is to just very slightly decrease the score of every segment after each.
Mutations are per chromosome or per segment.
Well, either way. I was just thinking whatever the smallest segment is in your chromosome. Like the thing is, yeah.
Oh,
I mean, this is all complicated because in real life you can also select on the mutations. So maybe a slightly more accurate thing to do would be, yeah. I mean, this is all very complicated. A really simple model of that would be you just every single chromosome, or okay, and even okay, sorry, simplest model. No, sorry. All right, I
yeah.
guess this is worth saying to give an illustration of how there's a lot of different choices. So the super simplest model would just be at the end when you're giving a score for the whole episode, you just give a ding, like a decrease.
Sorry. You talk about reward
Well, not
in another
necessarily.
setting.
Not necessarily. I,
So after crossover, the resultant chromosome gets its score knocked across the board. You're saying?
the super simplest model would be everything gets the score knocked down a tiny bit because in real life,
Okay.
and it's like the longer you go in terms of number of cell divisions, the worse the score is by a little bit.
Yes. Understand.
That might be fairly realistic, but that's already simplifying things away because, for example, um, there you could in real life you could freeze a cell line and then bring it back to life and use it to get DNA into your final cell, and then that frozen cell line would have divided less, and so it would have fewer mutations. So you can't just give it overall score ding for taking longer. You have to actually track the cell line. An even further complication would be so to solve that problem, you could just say every time you divide a cell, you slightly decrease the score of every chromosome or every segment in that cell. That would sort of fix that problem I said. But now there's a further complication, which is when you're actually doing the whole procedure, you get to select on which segments got more random de novo mutations in theory. Maybe none of this matters all that much, but maybe it does. It's like it's like something like, I don't know, 2 to 5 or 2 to 8 de novo mutations in a whole genome per cell division. So is that right? That's a lot. I think that's right.
This might blow up the state space, but on the whole, less than, let's say, 1% of our segments are affected by mutations, right?
Yeah, well, I don't know. It doesn't necessarily. Does it? Wait, why do you say it blows up the state space? I think we can just.
Because now every segment must also have an associated number, which is the number of times it has undergone mutation.
Well, no, I think we can just model it as the new cell line is not directly copying its segment values from the previous cell line. But it's um like if I let me see if I can draw this. The computer is being very slow, but so I guess my first intuition would be oh my gosh. Sorry. Thanks for your patience.
No problem.
So we start with these 2 chromosomes, and then we're going to induce meiosis, and we're going to get a new cell line, and the new cell line will have half of one of the chromosomes and half of the other one. And then the question is, what's the score for this new chromosome? So the obvious thing is that the score of the new chromosome is just like the score of the red piece is whatever the score of the red piece in the original chromosome was, and then the score of the blue piece was in the original one. What I'm suggesting...
Well, what adds or subtracts in the Brownian motion model?
You're saying we add or subtract a little bit from each one? Or is that... Or what?
No, the way I modeled, if I understood correctly, a score is given from the beginning of the chromosome until its end. And each segment adds or subtracts a certain amount to this score. So if we were to cut it at some point, we'd have to take all of the additions of the segments.
Yeah. So what I'm imagining in all of this is that at the beginning, as like a general parameter, we just for the whole entire, in a given like setup for the RL, we're just going to have a fixed number, which is like how many segments we're subdividing each chromosome into. Like it might be 10 or 100, or it might be 10 or 20 or something. And then that's the number that we use everywhere. And we just assume that crossovers happen at that point, and we don't assign values to... We assign values to the 1/10 segments.
Yes.
And we don't model them as being made of subsegments. Yeah. So like we would start at the beginning. We would have this population of 2 chromosomes, and then we would have a score for the first blue segment, a score for the second blue segment, a score for the first red segment, and for the second red segment, and then we would... Yeah. I'm not sure if I'm answering your question or not. Do you want to ask it?
Yes. Yeah. No, it's correct. Thank you.
Okay. Yeah, like at the beginning, the starting population, the values of these segments, I assume, would be drawn from a Gaussian, because they're being modeled as... There's some like highly polygenic trait, so it will have... The highly polygenic trait will have... within the first half of the blue chromosome, there's like or within the first half of this chromosome, there's like a bunch of genetic loci that are relevant to the polygenic trait, and so like by the central limit theorem, it's roughly a Gaussian. The polygenic score for the first half segment of the chromosome is like roughly Gaussian. Does that match your intuitions? I guess you're frozen. Are we back? Hello?
Sorry. Can you hear me now?
Now I can hear you. Yeah.
Right. Sorry, I lost the last 15 seconds.
Okay. Yeah. Well, I was basically saying we model the segment, each, like the segment, the first half segment, let's say, we model it as a Gaussian where that's coming from the assumption that we're looking at a highly polygenic trait, so it will have many genetic loci on the that are relevant to the polygenic trait. And so by the central limit theorem, you have like a bunch of coins, maybe they're biased, but you add up a bunch of small valued coins and you end up with roughly a Gaussian. So the value of each segment is basically sampled from a Gaussian, which is sort of the Brownian motion you're talking about. Yeah, and then, so what I was saying earlier is like one way to account for the mutation. You were saying like a full way to account for the mutation of cell division would be whenever we make a new cell, either by meiosis or by just culturing it. Like we also like, so we do meiosis and we get this like haploid cell with a crossover. Then we also are maybe culturing the haploid, so we get like another copy on the next day. And each time, and so your suggestion, if I understood it, was like we'd have to keep a counter on this blue segment here, which would be like, which would say 4, because it's gone through one
On
day
the
of.
red, on the blue, and on the red, yes. Every time it has mutated, if it you're saying that every operation we should counter it as it's a possible mutation. I was thinking only when mutation happens to increment the counter. I'm assuming physically it can be tracked when a mutation happens.
Yeah. And then so I guess I was suggesting we don't have to keep a counter. We can just whenever we make a new cell, we can, instead of saying the score of this blue segment is exactly the score of this blue segment, we just say the score of this blue segment is the score of the previous blue segment plus a small Gaussian.
Okay, so the Gaussian is from the
Or
mutation. Okay, I
yeah,
understand.
or sorry, maybe it shouldn't just be a Gaussian. Maybe it should be. I guess it would be like a drift. It would be like a Gaussian with mean negative 0.001 or something.
Yes, because it tends downwards. Yes. Mm.
And then I think that means the state space stays just like each cell has a vector for its chromosomes.
Yes. Yes. An array. Yes.
Although, yeah, then you do need to
The
then you do.
mutation that you're saying this model, it would not influence the state space. It would influence transitions.
Yeah, although now that I think another moment about it, I think you may end up being right that the state space. I mean, there's a bunch of choices we can make here, but the thing is, the state space. Really what happens physically, right, is like as you're doing this, as you're culturing your cell here, you're not just getting copies, you're getting a larger culture of more cells where like um, every single cell has a very slightly different mutation, right? Like you end up with, like a slightly different thing, if you see what I mean.
Yes, I don't think this should be a problem with modeling if the state space is a very large matrix which takes into account each possible segment and its values, then there shouldn't be any special difference in this case. So again, I think the only problem is in transitions, and ideally, hopefully in the course of learning the RL mechanism, we'll pick up that this is happening, that there's a slight downward shift whenever it does an action.
Hmm. Yeah, I mean the issue. Well, it's not really an issue. I mean this is a very minutiae sort of point. But I guess the thing I'm saying is something like part of the benefit. It's all very complicated. You could imagine. So you could imagine you make a haploid cell, and then you culture it for a while, but you end up with some cells that are sort of fairly well preserved and some cells that got a fair amount of mutation.
Well, the mutated cells will have lower values for the score.
Yes. So you can in principle do selection. You could make some colonies and like select the better one. But if we want to model that in the RL, then we can't model this cell line as just being a single like chromosome. We have to model it as like a set of cells, and that just complicates everything. So anyway,
Yes.
we shouldn't do this at the beginning, but I'm just illustrating another way that the state space might want to change and become more complicated eventually.
Do you think mutations could be that big of a problem?
Yeah, I think they are. I'm really unsure because I'm not a biologist, certainly not a stem cell biologist. We tried for like a little while to get numbers, but the very rough estimate is it's something like a 1 2 to 5, 2 to 8, 1 to 8 mutation de novo mutations per cell division. So
Uh,
your
If
so
yes,
your gamete, like if a person makes gametes, if a man makes sperm, an adult man's sperm will have many mutations, like maybe 50, 100, 200, something. I guess it depends on his health and how old he is.
yes,
And this is the main source of mutations in a child. The main source
yes,
of de novo mutations is mutations in the sperm. Um,
yes,
and so if we're having one or a couple of mutations in our cell line per division, and if you imagine like you have to culture a cell line in order to sequence it, which is usually true, basically. I mean, it's not exact. Yeah, this is all very. In theory, you can use it's like single
comment.
cell sequencing. Oh,
Understand? Can I make a comment about this?
yeah,
If the RL algorithm is efficient enough, it will tend to minimize culturings and divisions. Any sort of operation in training, there will obviously be a negative reward for any action because it takes too long. So, in theory, if the solution is efficient enough, this may only be a minor problem. This is something that we can check actually if we ignore mutations for now and see the best solutions produced by the algorithm, how long they take the entire process.
yeah. I take your point. And I do expect, based on some simulations, although I'm very, very far from confident, I do expect that it's quite doable to actually make a cell with a genome that you like without too many generations. But I also think that it is not a trivial constraint because, like, if you like do some envelope math, like let's just say you need to culture each cell line for 2 weeks, just to have enough cells to do the operations and to sequence and whatever. And that might be optimistic, but even if we're being optimistic, let's say it's 2 weeks. So let's say it's 2 weeks per generation, right? So that's already 20, 30, 40 mutations. That means if you take 5 generations, you're already sort of like at the level of mutation. You're already at like more than 100 or 200 mutations.
Yeah.
That's like
Mm.
sort of like, I think that's something like if an old man is having a baby, is making sperm, or like a middle-aged or older man. So it's already bad after 5 generations, potentially. And it might be
Mm.
worse. It's possibly it's significantly worse because the cells that we're operating under would be under stress because we're inducing meiosis and then we're fusing them. That's probably stressful. Probably quite plausibly makes their mutation rate higher. So I think it's a real
Yes,
constraint. It's like you have to do like a few. You want like it matters whether you do 3 generations versus 7 generations or something. That's why the
of course.
parallelization matters a lot. Yeah.
So this can be emphasized as minimizing the number of generations in a parallel setting.
Yeah. Um,
My poorly formed, I admit, intuition would be that getting a relatively low number of generations with this parallelizing should be doable. But of course, we'll have to see.
mm,
And of course, the framework should more than likely either include this from the beginning or be able to include it later.
yep. Um,
So again, transition of general score decrease is reasonably simple.
yeah. Yeah. That seems right. Um, I think
I don't wonder if this could lead to weird behavior by the algorithm to try and induce mutations somehow, because with that Gaussian, there's still the possibility of positive mutations.
yeah. I think that would be true. It
That
depending.
and I've read some studies I can't remember off the top of my head, which also use simulations that claimed that what positive mutations appear tend to be of tend to be more useful than the deleterious mutations are bad to compensate for their lessened frequency.
Hmm, interesting. Yeah, I don't know about that.
But
Sounds
I
strange,
think
but
it is it was only a somewhat speculative simulation study, but yeah, it would it would have explained why humans haven't died. Humanity haven't hasn't died 100 times over due to accumulated mutations. Mm.
um,
I think this is,
one.
however, an excessive amount of detail currently.
Yeah, yeah. All of this, probably almost all of this stuff that I'm saying is too detailed, but it still feels good to talk about, I guess, because
Of course,
well, it's sort of yeah. I mean, it matters in terms of the eventual application to actual scheduling. Yeah, but yeah. So in terms of in terms of like actually trying this out with RL, I worry about. Um, well, I'm curious if you think this is a problem. Maybe it's not at all a problem, but I worry about the state space being too big, even with like pretty simplified assumptions, just because the simplest way of doing things to me would be never discarding a cell line. I suppose you could just discard cell lines every generation, but that I don't know. I think that might be significantly worse. Like, I think you probably want to keep your cell lines around usually, or at least keep some of them around. So that means there's like a large actions. There's either a large state space where you every generation you're just adding more and more cells to the state. Well, yeah, I guess you're making the action space really big because you're like it's like you keep adding squares to the chessboard or whatever. It's like you
Yes.
if the action
I
is
would.
like the action's like pick any cell line you have to do meiosis or something. You keep adding cell lines, so you keep adding more and more actions.
Yes. I haven't looked into this problem very thoroughly yet, though of course it's relevant. My intuition was if our algorithms can play StarCraft or something very complex, it should be able to handle this.
Mm-hmm.
Uh, hmm, Discarding may be useful, especially in the later stages of the algorithm where it sort of has found its important chromosomes, let's say. Mm. There's also the fact that this discarding of cells I'm asking if it could be modeled as someone having already sequenced some of these cells and decided that they're probably not going to help very much, like they're very near the too near the median for the score or even below it. So even if there is some very large segment of very large score, it's very unlikely for it to exist.
Yeah, I think in practice you definitely could get cell lines where you're like, there's no good reason to keep these around. I do think that that would be it's a bit complicated. Like I don't think it would just be about the score because let me give an example. So suppose that hold on. I was trying to modify my Google drawing by clicking on the Google Meet uh, screen shared version. Okay, so how do I want to do this? All right, so let's say that we start off with these two chromosomes, the bluish one um, and the reddish one. And let's say the bright colors are better. Um, wait, sorry, let me. Hold on.
What I've mentioned with the discarding of cells, I was thinking of not not an unreasonable threshold, but one which will still give a lot of cells with chromosomes around the median, but you just discard those with obviously low scores.
Well, yeah, the issue is what I'm saying is that if you, yeah, let me try to work this example out. Sorry, one sec.
No problem.
Um, sorry. Okay, um, so we start with this reddish one, and the pale red is lower scoring, and the bright red is higher scoring, and then the sort of
Yes.
marine blue is worse, and the brighter blue is good. And now
Yes.
suppose that we run meiosis here, and we get this chromosome. So this chromosome
Yes.
sucks, right? It's got a low scoring um, greenish blue here and a low scoring pale red here, and it only has a small sliver of the blue. So probably we want to discard this. And if we are maintaining the previous chromosomes, then we do just want to discard it. This chromosome is better in every way because it just has strictly more target segments. However,
Yes.
if we have discarded this chromosome and we have not preserved this little segment anywhere, we might want to preserve this thing because even though the whole chromosome is much, much worse than all the other chromosomes, it contains a piece that would be part of the best chromosome. So it's not. So this is sort of an obvious point, but I just it's worth spelling out. Yeah, like the function of whether you want to discard things is like a whole function of like which pieces do you already have in which places, and yeah, there's like a general goal of like maintaining diversity or something.
Yes. I remember. Yes. Mm,
I mean, so that's like a thing you could leave up to the RL algorithm, although that's like another thing you're adding to the action
yes.
space of like you can also discard cell lines. Yeah.
Mm, yes. An action for discarding, which should it give reward?
Well, I think there would
Mm,
be. I imagine in real life there is some non-zero cost to maintaining a cell line, but it might be extremely small because I don't know how this works in real life. It might just be to maintain a cell line might just be like throw it in the freezer and there's plenty of freezer space, and you've already gone to the trouble of getting a monoclone and sequencing it in real life, so you would probably almost never want to actually discard a cell line, but you might
no, I'm
conceptually
no, I'm
go.
saying discarding from the point of view of the RL algorithm. It
Yeah,
discards, then it no longer takes it into account.
yeah. But I mean, you might not
Mm-hmm.
need to do that. Like as long as I mean, if we're doing like some machine learning here, hopefully it would just if it's obvious to discard it. Like whenever we would discard it, it should just be obvious to the machine learning to not use it, except in the rare cases where you actually do want to use it.
Well, I could say if literally every segment is negative, discard it.
Yeah. Okay.
That can also be baked in into the transition. So the RL algorithm doesn't have to do anything. Just if it is every segment is negative or below a threshold, discard it.
And this is supposed to be like an efficiency optimization where we're making
The
like
matrix will,
the policy evaluation is cheaper because there's fewer actions.
yes.
Yeah, I mean, I would have to defer to you. I guess my guess would be that even just the the inconvenience of you or someone having to add in that code is already not worth the gain because the gain just wouldn't be that big. But I'm just guessing. I don't know.
I don't think so.
Okay.
It shouldn't be. The state space, again, this is ML, everything is a collection of matrices, so simply discarding some of them, no, discarding some row or some column is not difficult, and it will make the job easier for the algorithm.
Do you? I feel a little bit confused about how these things work where the state space, where the action space is not like fixed, or it's like changing? Is there some standard way of dealing with this? Does that make any sense?
Yes, I understand. So instead of like in a tire game, the action space being 10 buttons that you can press or not, the action space being this is a point on the screen, and in each individual point you can do something. So you
Well,
have
but
a large matrix, and in
even that
every
even
place
that's
you can do
even that's like if you're playing like a video game, that's like a very large but fixed action space. I guess you're saying you
well,
could.
it's still fixed because there's only a limited number of operations that can be done. It's not continuous in this instance.
So in so okay, in our scenario, I guess you could say each turn, like each, like if you have, okay, I guess I'm thinking in terms of really big actions where like an action is like, what do I do for this whole generation? Like what's the whole set
Yes.
of pairings and
Well,
meiosis? But you don't have to do it that way. You could instead be like at each time like each action is like either add another operation to do in the next generation, like you're building up your plan, or press
yes.
go and do the whole generation. And that's just, so that's a much smaller action space, but it's a much longer episode, like many more actions.
Yes, I haven't looked too deeply into how these things are stacked per episode, but I'm certain I will. This seems like a type of problem that solutions exist for.
Okay, yeah, that's something I wouldn't know how to deal with.
Uh,
So it would be,
I'm
so
sorry if
your
it
expert.
sounds. I'm sorry. Sorry, please.
Oh, I was just saying, yeah, your expertise will be appreciated if you end up dealing with that problem.
I'm still a bit early in RL, but my intuition is if something as complex.
Sorry, I'm here. I was just getting my tea.
No problem. My intuition is if it's something as complex as an RTS, which could, I'm sure it has both a state and an action space larger than what we are dealing with, has solved these problems, then it's possible. And also, the way I thought it, maybe it will work, maybe not, severely penalizing doing an operation on the same cell or the same chromosome or whatever would mean that there's a bunch of sequential actions, but because they are each for a different part of our whole group of cells, they can all be done in one generation. And with that large negative reward, it would ensure that the point where the algorithm has absolutely needs to do an operation on a cell, a chromosome which has already been manipulated, we can say that is the end of generation. Whether this needs to be formalized somehow in the algorithm or if it's just the very large negative reward that I have told you, I'll have to check. But in principle, at least for me, it's doable.
Okay, I'm not sure I
Just
understand.
telling the algorithm to parallelize as much as possible.
Okay. Um, I guess there's also, maybe this isn't a problem, but there's a weird thing that I don't know how to think about, and maybe it doesn't matter, but actually there's two things. One of them is, or they're sort of the same thing. Okay. If you think of the, if you think of the, um, if the agent is building up, is taking a long series of actions to build up the like single big operation to do in a generation. There's um, there's a huge amount of like redundance, or there's many. It doesn't, the order of the actions doesn't really matter as you're building up the big action. So there's a huge set of actions
Yes.
that are all equivalent. That's a little weird. Maybe it's fine, but
Well, at least from my perspective, if I'm doing something with a group of cells, I'm doing something with another group of cells. Maybe right now they won't intersect, so it should be fine. Of course, I imagine there's also a relevant constraint to not do many worthless operations, but minor negative rewards for just any action can easily be added. So
I think what I'm saying is,
minus, let's say, please.
well, I think one thing I'm confused about is like, if we're doing the building up an action thing, then I think you have to include in the state space the previous actions that you've been adding for the next generation. Do you see what I'm saying?
Yes, marking has this chromosome been manipulated in some way? Yes.
Well, okay. So that here maybe I just disagree with you. I think you don't necessarily. I think you don't want to say that each cell participates in at most one operation, because, um, well, yeah. So if you have some haploid cells, you may have, um, let me illustrate.
If you're saying that each type of operation has into a generation, then this
Wait, sorry,
can
wait, Raresh,
be
sorry,
implemented with different,
wait, hold on. Can you hear me?
of course.
Sorry,
Yes,
you were cutting out. Can you just start over with what you were just saying?
of course. If you are saying that each particular type of operation has a different time cost, and that in one generation, for instance, you can fit multiple actions of another type, then this can be done with the negative rewards for each individual action.
Um, that's another good point, but that's not what I was saying. What I was saying was, suppose we have these three cell lines. Um, in a single generation, I might want to make a fusion of uh, these two chromosomes, make a fusion of these two chromosomes, and make a fusion of these two chromosomes. So like this guy is participating in two different fusions. So it's not true that if a
Yes.
cell line has one operation already queued up in the generation, then it's out for other operations. I think we need to allow. I don't know if you. I thought you were saying that we should disallow that.
It's less disallowing. You have to manipulate it with the reward because minus 1000 reward, it's not an explicit disallowal. The algorithm can do this action, but it will incur a negative reward. So it will try to avoid it as much as possible. Something like this can be done by lowering the negative reward taken by doing this operation again.
Yeah, I don't know why there. I don't think there should be a negative reward for doing an operation with doing multiple operations involving a single cell, or it shouldn't be a very strong negative reward.
I'm just to be clear, a generation. What kind of actions constitute a generation? You said the culturing cell, right? That's a generation.
Um, that's a good question. I don't know. I think there's a lot of choices one could make. Um, I'm trying to think about. So yeah, the thing is, in real life, there's no such. If you're actually doing this in a lab, there's no such thing as a generation. You just constantly are taking actions. You're constantly culturing cells, fusing cell lines, inducing meiosis,
Yes.
sequencing. You're just doing all these things every day. I imagine that it would be good to simplify this very much, at least at first, for the RL, where we just. I think. Let me try to spitball a simplified version that still is useful to do RL on. I think you would want to. Um, I think you would want to basically consider cell fusion as being free, and then you would consider that all of your cell lines are haploid. So the state space is a set of haploid cell lines. And if we're doing
Only
the
have
single,
one.
if we're doing the chromosome thing, it's just you have a set of chromosomes, and each chromosome represents a
Yes.
cell line with just that one chromosome. And then we are considering that the fusion step is completely free, which is not true, but we're just pretending. Um, and then in a gener uh, in a generate. Oh, were you going to say something?
I just wanted to say that I wouldn't consider even something theoretically free to be free for the algorithm, because any action, it will take more time, but not just time. It may let the algorithm just do stuff randomly uh, without reason, and this is bad. Needs to be generally a small time like constraint to make sure the algorithm actually does what it's supposed to and doesn't meander, and it has some pressure to develop efficient solutions. So even if in this case you would say fusion is free, okay, still a small penalty for doing it. So the algorithm knows that it needs to keep the amount of fusions to a minimum.
Okay. Well, I guess I was imagining that you would. Yeah. Well, I think you would just. I think you would. I think that would. I was imagining. Tell me if this is correct. I was imagining that that would all just be wrapped up in. There would be a cost to. Or sorry, the thing I'm the 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 model I'm describing is where in one generation the you take a set of actions where each action is produce a random meiotic daughter from a pair of chromosomes. And
Yes.
you can do multiple meiotic daughters from the same pair because you might want to do that, right? Um, so you just like the set of actions you take in one generation is a big like multi-set of pairs of existing chromosomes. And then but you also
Is this
and
a
then
fixed multiset?
say again.
Is this a fixed multiset in number? Only this number of possible uh, fusions.
You could do that. I was assuming that we just penalize the number, or we just
Yes.
each one every single the you get penalized by the size of the multiset. Just each operation is representing like a cell fusion and inducing meiosis, and then culturing and sequencing. So this is a very
Yes.
oversimplified model, but that's fine. And then you just like in real life we're overcounting the cost of fusion because we're saying you have to fuse again for each whatever it does it's yeah but you penalize for the size linearly and the size of the multiset Or yeah, you could just have a limit on it if that's better.
Okay. This penalization should capture a lot of what you're saying. This penalty is a general captures all of the efforts that are done in that particular step that maybe the model isn't explicitly aware of.
Yeah, and it's not a very good model of the cost, but it's fine for this simplified model. Yeah.
Right.
Um, yeah. Oh, I guess I wanted to ask you. I guess this is just a different topic, but I wanted to ask you sort of what the point is. What is the point of multi-objective reinforcement learning? Like, why can we not just wrap everything up into, like, it doesn't have to be a linear combination. It could be some other combination of of your different costs or rewards. But why does
Oh, of course.
what does it
Yeah.
mean when we have a multi-objective? Yeah.
Well, you can absolutely do that. There are quite simple wrappers which do exactly this. They turn a multiple objective problem into a single objective problem, where your objective is just a sum of some other objectives that the network doesn't really care about. The thing is that in certain problems, this is not workable. There's also the fact that in with multiple objectives, there is a Pareto front of solutions which are good for both parts, for both or all of your objectives, and they're in essence all equivalent mathematically, but subjectively the person making this solution might wish to emphasize this differently. And a lot of multiple objective algorithms, they don't give you one solution, they give you multiple solutions over this front, and you, the person making it, are supposed to pick one of them. Also, there's some mathematics, there's some classes of problems which cannot be represented with a single objective, though I'm not sure if that's the case here.
Okay. I don't understand the last part, but the first part made sense, I think. Or so like,
There was some papers like this may be not relevant here, of course, but there's objectives like the agent in a game should be able to, no, not it can, it does, no, the agent should be able to. This type of formulation, you can't really do it with a single objective. This is a mathematical result, so it's fundamental. Uh, it may not be relevant here, and initially I think that a simple single objective RL, where the single objective is some linear combination of the aspects we care about, should be okay.
um, gotcha.
Also, it's a bit more readable. My particular thesis will be on voting algorithms applied to multiple objective, which might be interesting, but it's not horribly transparent, and a linear combination is more transparent.
Hmm. So let me check that I understand. So, well, I guess I would. Is it fair to summarize like the main or a main reason, a main like abstract reason for multi-objective is basically the actual, okay, maybe I can't summarize it. One reason is that you basically don't necessarily know all of your preferences, or it's easier to know the dimensions that are relevant for your preferences without necessarily knowing your actual preferences. Like if you're showing a human, I mean, like for example, when if a couple is doing polygenic embryo screening, they might not want to try to write down in advance all their trade-offs between like how many IQ points for how much Alzheimer's risk or how much risk of bipolar disorder
Yes,
for a
yes.
decreased diabetes, but instead you just give them the whole, you give them all the dimension, you give a multi, you give them a vector, and then they use the actual vector to like gain new information about their own preferences, so to speak.
Somewhat. That multidimensional vector has a surface, which is the Pareto front, which is a way of saying the best for every objective. In a sense, there's no absolute maximum.
Right.
And you give them multiple solutions of the algorithm, yes, multiple vectors in the end for each score. And they look at them and they pick which one they think is more reasonable. So they might say, oh, this one has very low, uh, it has a very small reduction in the risk of schizophrenia. But we know we have a history in our family, so I don't like this one. We should look at another solution, which this dimension is higher.
Um,
Uh, if you're going to ask about details of how these solutions are produced, in general, multi-objective frameworks give you multiple solutions. It's fine. I'll um, have to look a bit harder at the details myself, but this isn't an additional programming or whatever constraint.
okay. Um, I also, so I want to say something abstract or sort of zoomed out about the whole problem, which I guess you're probably familiar with, but I don't know the correct vocabulary if there is one. There's sort of like several levels of let's see, how can I put this? One question is I don't know exactly what is the type signature of what we're training, because so you could have you might have a specific couple or a specific parent that you want to apply iterated meiotic selection to those cells, or just in general you might have a specific set of cells, and furthermore you might have a specific utility function on genomes. So if you have a specific set of cells and a specific utility function, that in itself is a non-trivial, like the task of doing IMS, given that specific starting set and that specific utility function, that's already a quite non-trivial scheduling task, which you could do reinforcement learning to, and then what you would be because of the stochasticity that when we do crossovers and stuff. Um, so you could train a model which is I do well on this specific set of cells and this specific utility function. Or in a more multi-objective way, you could say I don't have I do have a specific set of cells. I don't have a specific utility function, but maybe I have a set of like 10 scores. That's my multi-objective. And then I want to do reinforcement learning that produces a good spread of the Pareto frontier or something.
That's
Or
uh, that's inverse reinforcement learning,
okay,
where you're given the 1 second. When you're given the solution and the algorithm is supposed to learn it, that is different. I think that, well, it's doable, though it's a bit of a different class of problem.
Wait, I'm not saying that the algorithm should infer. Wait, I thought inverse reinforcement learning was. Or what's inverse reinforcement learning?
So reinforcement learning, you want to maximize something. So you want to get the maximum possible score, let's say, for a particular environment. Well, in inverse reinforcement learning, you already know what you want, not the maximum, but a specific motion, let's say, or something, and you go backwards. So instead of going forwards from the rewards, you try to make the algorithm learn what the rewards are supposed to be to arrive at a certain point.
Okay, yeah, I don't think I was saying that. I think I'm just saying. Uh, I think what I'm saying is, in real life, you would want to. In real
Actually,
life,
I have a small critique of this scenario. If these solutions are below the Pareto front, which can be calculated mathematically, then I see no reason why you shouldn't just give the one with the higher scores all around. If it's outside the Pareto frontier, then the algorithm has not cannot produce it, or is at least to a degree underpowered. I'm not sure how useful this would be, and also I'm not sure in real life with a couple wanting this if they would phrase it in such a way. To me, at least, not even a vague "I care about this 5 times more than I care about that," which could theoretically be modified by having that linear combination be different. Mm.
I'm not sure I'm following. Um, let me try to say the scenario again. I I'm not sure I'm. Yeah, we might be talking past each other.
So, please.
I think what I'm saying is like, in real life, there would, no matter what we do, there will probably be a significant amount of stochasticity, meaning you won't end up with like the optimal genome according to your,
Of
even
course,
if you had, even if you had preferences. So,
yes.
um, uh, and you might not, and the parents may not have even tried to define their whole utility function. So for both of those reasons, um, you're not necessarily trying to. It's sort of multi-objective, or let me say it a different way. The actual thing you want is for the procedure to be likely to produce at the end a set of several cells that are, in some sense, a good um, reason, somewhat well populating the Pareto frontier, if that makes sense. Like
So the
you're like,
solutions to be spread out and not concentrated in a small region.
yeah, and to be, yeah, you want them to be, yeah, you want the solution, you want the Pareto frontier of your final set to be fairly spread out. Yeah.
Hmm. I think
Like,
this is, I'll have to check, of course. I'll have to see. This may be a function on the general stochasticity of each individual solutions, how it picks out its actions, because we'll probably may have a bit of randomness in each action, of course. Mm. I'm not sure this will be that large of a problem, because even if we have three scores, let's say, the frontier should be very large, inherently quite large. I doubt it's likely that a lot of solutions will be very narrow.
Well, what I'm saying is like it's a different. If the aim of an actual run of the procedure, if the aim is to produce a single cell line, that's a different aim from the aim of producing a set of cell lines. It would make you take very different actions, and
Yeah,
you want to produce a set because, like, a way to model
yes.
this would. Okay, yeah.
From what I saw, I have to look at the implementations more closely. The ones that take this into account already produce multiple solutions.
Okay.
Again, there is no additional effort required. And each solution will be, well, the list of actions that were taken.
No. It's the
I'm sorry if I'm not comprehending correctly.
I think I'm just not communicating well. Sorry, I'm just frustrated that I'm not, because I don't have the ideas clearly in my head. Um, like, also I might be misunderstanding what you're saying. Um, I think that
Are you
the
talking about the difference between how the algorithm is trained and how it is deployed during inference?
I don't think so. What I'm trying to say is that when you're saying a solution is found, where a solution is a sequence of actions, what I'm saying is we need to find a single when we actually apply the thing to actual cells, we get one sequence of actions, right? Like, I mean,
Yes.
you could go back and run the whole thing again, but that's very laborious. We assume we just want to do it once. Um, so the thing we're looking for is the algorithm is supposed to output just like one sequence of actions to start with some cells, and then at the end stochastically produce some other set of cells. Um, what I'm trying to say is the um, the actual utility function shouldn't be like the actual utility function is something like um, there's two different ways to say it. One way to say it would be the actual utility function is, do you have you pushed far on the Pareto frontier, like gotten pretty like have you pushed up along many dimensions, so like gotten high scores
Yes.
on many dimensions, and also have you covered a fair amount of that Pareto frontier. That's the actual utility function that is applied to a single set of action, a single sequence of actions. A different way to say that would be um, the actual utility function has stochasticness in it, I guess. Like, it's sort of like at the end, like the parents look at we run we do a bunch of stuff with cells, and we're like, okay, here's like 100 different cell lines. They all have like pretty good scores, but they have different scores, and so there's none of those trade-offs. Like, it's a big Pareto frontier, and then the parents like think about that, and they're like, okay, here's our preferences, and there was information in that final set of parental decisions. There was information there that we never had in our that the agent doing the scheduling did not have access to. Does that make sense? So both for the
So,
training.
gi- so giving possible solutions to the parents, and when they say I prefer these solutions, we modify the utility function to resemble it.
Well, but we don't get a second chance, is what I'm saying. In real life, you would just want to. Maybe you get as much information from the parents as you can, um, but you can't get all the information. They're going to end up making, like, they're going to end up maybe changing their mind depending on what we actually end up with.
All right, I understand.
And so therefore what I'm saying is, if you have that like component of the utility function that you didn't get to train your agent on, and you didn't get to have that information when you were actually doing the cell operations, so you're going to end up with like a set of cells, and then only at the very end does that utility function get applied. In order, like, the actual strategy that an agent should follow to do well at that task is something like, when you actually run the cells things, you should actually end up with a set, and the set should populate the Pareto frontier fairly well. That way you get to take advantage of that way you're like helping the parents have more options to choose from, basically. Does that make any sense?
Besides,
We
as
maybe
the
maybe
given solution assumed that its entire state space was those cells which exist, the only way to run it multiple times on the real cells would be to have multiple copies of them. I think we're a bit stuck. So you're saying that we've run everything. We have solutions, written solutions, not actually executed on the cells, and the parents look at each result of the solution and say our preferences align more with this one.
you could also do that. That's not what I was trying to say. What I was trying to say was, what will actually happen is you won't be able to capture the parents' preferences beforehand. So you won't be able to train your AI system to actually optimize for the parents' preferences. And that's fine.
Well, there will be a standard utility function which takes each one of the aspects equally, let's say. And you're hoping that it will produce solutions which will, each solution will be in a different region, which each region emphasize the set of those objectives more of those scores than the others.
Yeah, so I think that would be fine. I think we don't need. The thing I'm saying is not something that we really need to worry about right now.
Uh, I'm not sure about the deep mathematics, but hopefully the set of solutions produced are spread out and not too close together.
I mean, it's just that
You're worried? Are you
if you if
worried that the solutions might be in a area of the frontier which does not capture the preferences of the parents at all?
the thing I'm worried about is just that the way I understand the basic thing, which I think what I'm calling the basic thing is, we just have like a single utility function. We just know the utility function. I think that's perfectly fine. I think that is still interesting. It still tackles the main part of the problem. We would learn stuff, etc.
It
So
can be modified with
yeah,
for other trainings. Okay.
but the thing I'm saying is, um, a structurally incorrect thing with that is that the actual thing I want, like what you're training the AI, what you're training the agent to do is to um, let me see. Okay, so suppose that okay, here, I think I can maybe illustrate this fairly simply. Um, sorry, just one sec. Okay, sorry. So this is supposed to be like, there's two objectives. We have a multi-objective thing. We have two objectives, and then here's
Yes.
here's a Pareto frontier.
Yes.
Now
That's exactly what's represented in the literature. Yes. With the example.
So now suppose we pick a utility function, and suppose the utility function is just x plus y. It's just the linear. It's just the sum of the two things.
Yes.
So there is there's an optimal point here, right? Which is
yes. The
which is here.
Pareto frontier is a general thing of trying to maximize multiple objectives, and the algorithms which produce it are not simple single objective reinforcement learning with this linearization. It's a case of more complex algorithms giving this. For x plus y, the optimal solution which it will find will be that middle point at 45 degrees. But if
Right.
we apply tailor-made multi-objective algorithms, which there are a few standard ones,
Mm-hmm.
they might give any solution across the Pareto frontier.
Okay, but so here's the issue I'm having. When you say that, here's what I think you mean. Tell me if this is wrong, because I'm not sure. I'm just guessing. I think what you're saying is you run this tailor-made algorithm, and then you end up with an AI that or an agent that sometimes it produces a point here, sometimes it produces a point here, sometimes here, sometimes here, sometimes here, or maybe you tell it a utility function and then it produces one of these points. Is this?
Sorry, I was not clear enough. So with simple linear utility function, or for with whatever weights we wish, in this particular case, it will try to get the solution of the value at the point at the 45 degrees on the edge of the circle. So there is one solution.
Mm.
If, however, you take a proper multi-objective reinforcement learning algorithm, which there are several, then it will try to various means, have to check more thoroughly, to maximize both. And if you say, please give me 3 solutions, it will give you 3 solutions on the entire edge, hopefully edge, of this circle.
Yeah.
So not just the one in the middle.
And when you say it'll give solutions, what does that mean exactly?
So the sequences of actions which produce one of the 3 vector values, yes.
Okay. And is this basically stochastic? Like if you run the resulting like you've trained an agent, and then if you run the agent, you will randomly get a point on the Pareto frontier. Is that right?
Well, because the state space that we have is known, uh, we can do either. We can make so that the agent picks things stochastically or not.
Okay. So so you have these two different objectives, the X and the Y, and then
Yes.
you run a multi-objective thing, and that produces an agent which, when you like run the agent, it'll produce
I wouldn't say agent in this case because this is why I was asking about training versus inference. In my mind, we have an initial state space, and we will train our algorithm on this initial state space, presenting all the cells, etc. we have. For another initial state space, other cells, other whatever, uh, if I think it would be feasible, at least in the early stages, to just train again. And at that point, we only care about an algorithm which has managed, of course, after a lot of iterations, a lot of training, to produce just a few solutions which work well on that particular trained state space. It may not be optimal if we change that initial state space, the algorithm that we have trained, but yes, it's there, and we clearly have our optimal solutions with their associated actions.
cool.
Not all RL is can be well, it can, but should be conceptualized as with agents.
Okay.
Because if it's agents, then yes, we could say at any point with this probability do this on this region, though I'm not sure if it's particularly useful in this situation, though I have to check this as well.
Okay. I'm not sure I understood all of that, but it's um well, what would you call the thing? So after you trained something on the multi-objective thing, given a fixed starting state, like a set of cells or something. What would
Yes.
you call the result of that training? You've run the RL multi-objective algorithm. What's the result? If it's not an agent, what would say solutions, or
Yes, it's given a set of solutions.
is the solution like one of these points, or like it's a set of
Well,
cells that
hopefully,
results
yes.
with the set of actions?
Hopefully, yes. Actions which are not stochastic. It's clear are the steps that it has taken to arrive at this point.
Okay, so these are basically like execution runs. Like I would say these are
Yes.
execution runs of an agent. But what does the for this algorithm, what is it using to produce the how would you apply this to actual cells? Like you'd run your multi objective thing, and then you get something, but now we actually have to do it with cells where the randomness is not coming from the simulation, it's coming from the world. So you can't use like the like if this is producing like a set of actions with actual randomness, actual
Well,
results of randomness that happened. Yeah.
of course, it still has within it a neural network or policy calculation
Okay.
thing, whatever. And of course, that can be applied to a real situation.
Okay. So it produces a policy. Um,
Depends on the algorithm. There are multi-objective variants for a lot of standard RL algorithms. both policy-based and not. So there is a multi-objective variants of Q-learning, and on the other hand, there are multi-objective variants of policy-based algorithms.
Okay. And is it a single policy that sort of does it randomly produce something on the frontier, or is it like you can like tell it what the actual utility function is, and then it will go to that point?
Uh, I, I'm not sure uh, if I've looked into this already. Um, I look. I've mostly for my thesis. I've read the theoretical works mostly, but
Hmm.
I'm just beginning to look into actual implementations. So
Cool.
I cannot answer it at this
Okay.
time, but I'll keep it in mind.
Okay. Thank you. So I think my problem still is a problem that I was trying to say earlier, where so we have this policy, but what it does on a single run of the policy, it takes some set of actions. There's some randomness that gets sampled, and then we end up with like a cell line on the Pareto frontier, and then
Yes,
we could run the policy another time and get another cell at a different point on the Pareto frontier.
yes.
So there's a sense in which we're sort of filling up the Pareto frontier in that if we ran the policy several times, we would sort of fill up the Pareto frontier, right?
If it's trained right, yes, it should.
So what this would correspond in the real world to is you run your whole big cell lab sequence of operations that's expensive, and then you get one cell line that's on the Pareto frontier. Then you do that all again, and you get another cell line. Then you do it a third time, and then you give to the parents, you give them those three cell lines, and you're like, here's three different cell lines, and here's their scores on various polygenic traits. Which ones do you want? But what I'm saying is that's extremely expensive because you have to do the whole big operation for
Yes.
each Pareto frontier cell line. And what I'm saying is there's a different sort of objective, which is that I want a policy which on a single run produces several different cell lines just within this one experiment. It produces several different cell lines which
Uh,
in one run kind of fill up the
yes,
Pareto frontier a little bit.
that is a separate objective, of course.
Yeah.
I have to think about how to conceptual, not how to uh, create a reward around this, because of course, like this, it's not simply maximum score gets bigger or somewhere. It has to be in multiple places. Uh, I don't see why this wouldn't be doable. Though I'm asking if you think this should be done at this early stage.
No, I think it's probably not worth any additional complexity. I think even just having a known fixed utility function that's just like x plus y or something, that's perfectly fine. That still captures a bunch of the interesting problem.
Again, that's basically single objective reinforcement learning. It's not truly a multiple objective.
Yeah, I think that for now probably single. I mean, if you want to do something with multi-objective, that also seems cool. But and doing the multi-objective thing, yeah, might enlighten us about this question and other questions. But I think even the single objective thing is already quite non-trivial and would tell us a lot of what we want to know. Yeah. Um,
Understand?
I think what I was trying to say earlier was one way that maybe you could formalize this or operationalize this is by saying the way that we evaluate a whole episode um, is by you you in an episode, like the policy produces some actions. At the end, it produces a whole set of cell lines, and then at the end, we randomly select a utility function. Now, I don't know what the distribution should be, but
I'm not sure if it's necessary. At the end, we have several solutions in the whole conglomeration of cells. There are multiple solutions out there, and they are presented.
well, I'm saying this is a way that you could convert this into um, having like a. It's a complicated utility function, but it's like a fixed one. I don't know if it's like a single fixed utility function. It's not like linear because it's somehow, I think it's like, so like you see these lines with like the different slopes that would like correspond to different linear combinations of x and y, right?
Yes, but bear in mind that for each of the slopes you are making, there's still only a single solution within the Pareto frontier. And the Pareto frontier and what's in it is what's achievable mathematically with the starting state space. So it's not simply where the algorithm goes with its solution. This is the mathematical constraint on the values of these scores, these objectives. So it's impossible, not that it's or whatever. It's mathematically impossible to have a solution outside of that circle.
Okay, then I might, yeah, maybe I'm trying to talk about a slightly different concept, which is, um, well, I mean, if you just, yeah, like if we just think about, yeah, I don't necessarily mean Pareto frontier in terms of what's mathematically possible. I think I mean, I'm just trying to say like, uh, basically, we're with this more like complicated thing that I was talking about, where you're trying to within a single run produce a set of solutions, so to speak, or like a set of cell lines.
Yes.
Like formally, now we've kind of we've made things more complicated, where the straightforward way to formalize this is your space of end states is like very, very high dimensional, because you have a dimension for every single objective and every single cell line, right? So it's very high dimensional. But I want to say, and then you could express things. Yeah, yeah, maybe this. I don't think this matters, and my thoughts aren't quite clear enough to. Yeah, we can move on.
Understand. Okay.
Yeah, I think we communicated about the main point. Thanks for bearing with me. So
No problem.
so yeah, what else should we talk about? Well, yeah, do you have questions or thoughts?
I'll have to revise both from your articles and the other sources the general process. What I wanted as a small answer, if the simulation I have done, pure IMS, let's say, each chromosome in vitro, sorry, if that is a usable basis, or if you believe that conceptualizing it with the diploid and haploid cells treating them as existing entities in the algorithm is useful to begin at that level of complexity.
I didn't read. I did look at your Python, but I didn't read it carefully. My guess is that a good place to start is this idea of the state space is a set of single chromosomes. The action space for a given generation is a multiset of pairs of chromosomes that are in the state space. And the transition function is each pair of chromosomes in the state space produces like a random produces a single chromosome that is a random that's it. There were crossover at a random in one of the 0.2 seconds, and a chromosome just like some global segment number k, maybe it's 10 or something. Every chromosome is just a sequence of 10 numbers, each of which is sampled from a Gaussian with standard deviation, whatever it is, root k or 1 over root k or whatever it is. Does that sound right, or does that make sense?
Yes, but I want to make sure the point of crossover is not chosen, it is random.
Yes, that's
so at any singular action, the only thing the algorithm can decide is which two chromosomes to do it to.
Yeah, and then I suppose, I think you want to have I suppose you could drop the whole generation structure thing, but I think it's kind of bad because the issue would be, I think you do want to have the generation structure somehow, because otherwise you may have like very high depth, if that makes sense. Like you may keep operating on the same chromosome. But basically, the simple thing.
Could you please repeat the previous 10 seconds? You're cut off.
Can you hear me now?
Yes.
I don't know where you got. I guess what I was saying was I think you want to
drop. It might be bad to drop the generational structure.
I was saying you probably shouldn't, and the reason is that I think without the generation structure, there's no disincentive to do the like straightforward thing that you had brought up about the linearly, like just do the first segment, then the second segment, then the third segment, then the fourth segment, because that is pretty efficient in terms of. I think that's like optimally efficient in terms of the number of cell lines, but it's bad in terms of the number of generations. It has a high serial depth, which is bad because of the mutation thing.
Of course. And just to make sure, each generation, you said it shouldn't be thought of as each chromosome having only undergone one action. So that's not sufficient to say generation.
I think you should allow multiple actions per chromosome.
How many?
Like there doesn't, in theory, there doesn't have to be a constraint there, or there. In theory, you wouldn't want a constraint there because if you imagine, if you imagine that it's that every generation is extremely costly, this is unrealistic. This isn't real life. This is unrealistic. But mathematically, if you imagine that a single generation is extremely, extremely costly, then you'd want to be extremely confident. Like, if you want to make sure that you have a pair of chromosomes where if they get the right crossover, then you're done. So you're going to pair them up, right? But you want to pair them up many times. Like in your multi-set, that
Yes.
pair will appear many times. And so you want to become extremely confident that you will get the crossover that you want. And in principle, if the generation cost is like exponentially high or whatever, the correct move could be a very, very large number of all at once in a single generation. You want to do 100 or 1,000 from just that one pair mathematically. But yeah, I think in practice, if you need it for the RL algorithm, then you can just say like, I mean, for example, you could say each chromosome, like if there's 10 segments, or if there's k many segments that we're dividing our chromosomes into, then it's going to be on the order of k, right? Like it could be like k or 2k or 3k or something, because if you sample k many, what's the chance? Like the chance that you succeed at getting the crossover you want is like 1 over e or 1 minus 1 over e or something like that. So you probably, depending on all the relative costs of having more cell lines versus generation time, you might want to do 2k many samples from a pair. You probably don't need to do 10k. So something like a small multiple of k is a fine constraint, probably, is my guess.
Okay. On the same group of chromosomes that has been chosen. So not in total. So if I
Yeah, the
have
same pair. Yeah.
okay,
If you're if you're
same isolated pair. So if I choose two groups, they can't overlap. Or if they overlap, that's another generation.
I didn't understand. Well, to be clear,
I apologize for my
oh no,
confusion.
I if I
I am
if I understand, I think no, it's fine to have different sets because these things are representing like a whole cell culture. So we have lots of cells.
yes,
So we can like take a chunk of these
yes.
cells and combine them with a chunk from another. Does that answer your question? Or
On the low mathematical level, can I recap
yeah,
a
yeah.
bit to make sure that I understood correctly? The state space is all of the chromosomes that we have, and the action space would be picking a number of them, let's say 5. And within this group of 5 chromosomes, between two of them, uh, is this picked or not? Can the algorithm pick the specific 2 chromosomes from this group?
Yeah. Oh, I wouldn't even. I don't we don't necessarily have to um have a separately selected set. I guess what I'm saying, an action, like one generation action, is it's just a set of it's a multi-set of any pairs. So yeah, you can pair anything with anything. I'm not. I guess I wasn't sure why you had the step where you're picking a set of 5 first.
It's how you phrased it, sorry. Okay, so the algorithm picks what chromosomes to recombine
Um,
to a step which represents the entire recombination operation, everything it implies.
yeah. Um, let me, I can write. Sorry, I don't know, my computer's so slow right now.
Okay, so I conceptualize it correctly. My, I think the fundamental question I have is in this whole process. What is considered wait, duplicates of the pair, not duplicates of the chromosome.
Correct.
So this crossover can be done on the same pair, but not from two chromosomes from each which already belong to the same pair, two different pairs.
No, you can. Yeah, there's no constraint on that.
So if I have C1, C2 in P1 and C3, C4 in P2, I can do in the same generation C1, C4.
Yes.
Okay, I understand that. I thought correctly. I think my fundamental question is how would you define a generation? Because from what I gather, the very simple solution I proposed, you said it would take many generations, but under these rules, with the chromosomes being allowed to be used between pairs, um, where the generation, what would it, how would it be defined with this set of rules?
Well, generation would be just a list of pairs of existing chromosomes, and then you would the result would be a larger set of chromosomes, which is all the chromosomes that already existed plus the ones that were produced by these pairs. So to implement your algorithm, which I'll just
So the state would grow, yes.
I'll just summarize your algorithm in case anyone's watching the recording. So Rareș's algorithm is you take
This algorithm assumes that crossover point can be known in advance. If it can't be, then the algorithm will not work unless it's modified to do it so many times until you get the segment you want.
Yeah, so the way you could do it would be in the first generation, you take the chromosome that has the correct segment 1 and the chromosome that has the correct segment 2. And then the whole action you take is just combine those two like 20 times. Let's say there's 10 segments. So we just take those two and we have that same pair repeated many times. And then so that means we get like 20 or 30 recombinations from those two chromosomes. Very likely, at least one of the resulting daughter chromosomes will have the first two chromosomes correct because it had the crossover right in that exact spot between the first chromosome and the first segment and the second segment. So that's in one generation, we've gone from the best we have is just the first segment being correct, and now the best we have is the first two segments being correct. So then we take that chromosome and then we apply that again with whatever chromosome has the third segment being correct, or by correct I mean the maximum value for that segment.
Maximum.
And again, in the second generation, we're going to apply this 30 times to just that one pair repeated 30 times. So we're going to get a bunch of daughter chromosomes crossing over, and then probably at least one of them will have a crossover between the second and third segments. And then we will produce a daughter chromosome with the first three segments correct. And we keep doing this. So this works. It's inefficient in a few ways. And one of the big ways that it's inefficient is that it takes a lot of serial time where you're, if there's 10 segments, it'll take you 9 generations or whatever. It is reasonably efficient in terms of the number of cells, in terms of the number of cells you produce. You could be even more efficient where every generation you just make a single crossover. You only produce a single new chromosome, and you do that way you can stop immediately when you get the correct crossover. That's even more efficient in terms of the number of cells, but it will take, like 100, it'll take like 50 generations or something. Yeah.
I think I found the source of my confusion. I treated each chromosome as essentially something static. When a crossover between the two chromosomes happen, we're assuming in that model that those two chromosomes that existed before no longer exist. It's just the new crossover ones.
We don't have to kill them. They can still exist.
Correct. That was the assumption that I made in my
you're assuming that when we combine them, they die.
Yes, that's why I was confused about the generation. So generation is if we take the output, let's say, of one recombination, those new chromosomes, and combine them either with older chromosomes or chromosomes resulting from another pair, that's another generation.
If you combine a new chromosome with either it's like other guys in the same age group or previous guys, that requires another generation.
Yes.
Yeah.
Yes. Okay, I understand now. sorry for taking so long.
No, yeah, that's exactly why conversation is good.
Yes.
I'm glad we figured that out.
Thanks for bearing with me. And such mutation rates are important because each new generation, the chromosomes will be successively worse than they would otherwise be.
Yep.
Okay. The two results of crossing over, correct?
Yeah, I guess that's right. I think that's a modeling decision that you could make to include both or just one.
Because ignoring it would imply discarding the rest.
Well, I mean, the thing is, like it's true that if you get one crossover, like in real life, if you did meiosis and you got one crossover, you also got the other crossover. But also in real life, we're not like getting every cell. We're getting like probably we're doing these things in big batches where we're taking like a whole bunch of cells from C1, a whole bunch of cells from C2, and then like mushing them together and merging them, and like a bunch of them die and a bunch of them don't merge, but some of them do merge, and then we do a process of selecting, and so we have a big batch of cells that have both of them, and then we induce meiosis, and a bunch of them die, and a bunch of them don't do meiosis, and then we have a small set that successfully did meiosis, and then we select for those, and now we have a big batch of cells that did meiosis, and then we select single cells from there to grow into a clone, and a lot of them die when we try to grow them. And then we have some clones, but there's no constraint that says that if you made a clone of one of these haploid cells with a single chromosome, so to speak, there's no constraint that you also happened to get one that was the opposite crossover.
I understand. Okay, so the crossover operation isn't, it's not outputting two chromosomes, it's outputting one chromosome mathematically. And but I see, no, it's not arbitrary because there will be different results whether C1 is the first one, quote unquote, or C1 is the second one.
Yeah, there's two K
So,
different. If there's K segments, then there's 2K plus 2 different crossovers.
so the algorithm would have to decide not only which pair of chromosomes, but which should be, let's say, the left side and which should be the right side.
Well, it doesn't. I don't think that the algorithm has to. It doesn't. The choice doesn't matter because, like it's random which.
But it has to be one or the other. Oh,
we can assign it randomly. It doesn't matter. The transition function can assign it randomly. I mean, yeah, the algorithm has. I guess if the algorithm's like picking us, I mean, if there's a if
Okay,
there's a
yeah, sorry.
if
Yes,
there's a way to
yes. Got
okay,
it,
if there's
got
a way
it.
to make things more efficient by making the algorithm only have to output a set rather than an ordered tuple, if you can make it twice as efficient, then
Yes,
that's
yes.
good, and that's fine because yeah, but whatever.
Okay, I understand. You mentioned the point in 0k. Does this represent that crossover just doesn't happen?
Yeah, basically, or the way I'm thinking of it is that like in real life, the distribution of where the crossover happens is sort of complicated. There's like hotspots, and there's generally like it's not just uniform, but even if we pretend
Of
it was
course.
just like if we pretend it was just uniform at random, even across a continuous chromosome, then um, that doesn't line up with our discretization where we just have k segments. So I'm sort of like, well, if it lands in the first like half segment, then we count that as a zero crossover. Yeah, I mean, I'm just not worrying about that too much. It's a very mild. It's somehow inaccurate, but it doesn't matter very much. Yeah,
Okay.
you don't
Just
you
from
don't
the point of view of sorry.
you don't need to include the zero and the k. You could say from 1 to k minus 1.
Okay.
Yeah.
I'm saying because from the point of view of the algorithm, that's just a wasted action that it couldn't foresee. It
Yeah.
doesn't change the final solution because if it doesn't work, you try again.
Yeah, but I mean, in real life, you might get a crossover that's very near the edge, and that sucks, and that could happen.
Yes, yes, I understand.
Yeah. Um, I should probably
So
get going soon, but yeah. What else should we
I
talk
have
about?
time. So from your perspective, this would be a reasonable simplification of the task at hand at this stage.
Yes.
All right, so it will have an expanded state space over time. Okay. All right. Uh, Thank you very much, Mr. Tilson, for bearing with me and my confusions. Again, I have read your works. Maybe I've misunderstood or misremembered parts of them, which is why maybe it took a while for me to uh, understand things you have said, and I apologize. I hope I'm at a better place now with this description that you have provided. And I think that even though the expanding state space is a challenge, of course, I think a reasonable, at this stage, single objective reinforcement learning algorithm is doable.
Yeah. And I mean, I somewhat defer to your your expertise and your experience with reinforcement learning, and if you work on this stuff, you will quickly come to know much more than I know about the specific domain. So I would defer to your knowledge about, like, maybe you should add in constraints on the state space, or like, yeah, you could try. It's not crazy to you could delete this. You could say all the previous set dies. You could say that. That's probably not what you would want to do in real life. It would probably lead to worse solutions, but maybe not that much worse if it simplifies things. I defer to your knowledge about how to make the RL work well. Yeah.
My knowledge is still building, of course, but now that the problem is more clearly stated, I go ahead with this. All right. I'm very glad for the conversation. I think that from my perspective at this point, I can do a better simulation with what you have done, and then after that simulation is done and you consider it appropriate, RL solutions can be begin to look for them.
Yeah, that sounds fantastic. Yeah, I mean, I super appreciate you, yeah, wanting to figure this stuff out and chatting with me about yeah, trying to figure out the problem. It's a good conversation.
You know, for me, thank you.
Thanks, Rareș. Yeah, have a good rest of your evening, and hopefully we'll talk another time.
Sure. Thank you, sir. And as soon as I have something, I will email you.
Cool. Thank you so much.
Thank you, and have a wonderful day.
You too. Take care. Bye.
Bye.
Is there anything I should comment on? So, yeah, I mean, there's just like a ton of different like dimensions that you could take this in. Like, yeah, I guess we discussed a lot of them, but you know, like, first of all, you can have, like, the set of chromosomes is supposed to be like, you have a set of cells. Each cell has a single chromosome in it, but of course you can have like an actual genome. You can model the time and other costs of merging cells, isolating cells, selecting for haploidy or diploidy, or euploidy. You can model those costs and the costs of sequencing. You can model whether or not you have sequenced things or not. You do not necessarily have to sequence things. You also, if sequencing takes some time, like if it takes a day or two for like a SNP array, or whatever, or 3 days, you can already start doing actions without knowing the sequence, and sort of like a branch prediction in a CPU, where like you're branching on, like, if the crossovers happened the way that I wanted them to happen, then I want to do these pairings, but I don't know yet whether they happened, but I will just pretend that they did happen the way I wanted, and proceed as though they happened. And then if I find out later that actually they didn't happen, then I can go back and fix that. There's also like time travel sequencing, where you or just time travel in general, where by time travel I just mean like freezing a cell line, although I don't know how damaging freezing a cell line might be. It might be fairly damaging. You can also, of course, as we discussed, you can model different de novo mutations. Then, of course, there's the whole question of different crossovers, where, or of multiple crossovers, where there's not always, I think there's not always any crossover. Sometimes there are 0 crossovers. There is also often 2 or 3 crossovers, or even 4 in a single chromosome during meiosis, like in spermatogenesis or oogenesis. The how many crossovers is different for different chromosome numbers. The positions of the crossovers can vary. So like, you know, we have a picture somewhere. Yeah, so this is from Bell et al. 2020. You can see that. So this is saying this is the position on the chromosome. So this is a chromosome that's I guess about 200 megabases. Chromosome 5. I think the gray line might be the centromere. Don't quote me on that. Maybe this is... No, maybe the gray line is right in between the maybe this is the centromere, and these are the telomeres, and then here's like the heterochromatin, like where most of the functional DNA is, like in these two patches between the centromeres and the telomeres. So that's where you're getting, I guess, fewer crossovers, apparently. Maybe I'm just wrong about these positions. Anyway, depending on where you are in the chromosome, there's different crossover rate. It's also hotspots, and it varies between different chromosomes. And when you have multiple crossovers, they're not independent. They're not in independent positions. There's some correlation, whatever it is. And here you can see that there's there's quite a noticeable fraction, depending on the chromosome number. There's quite a noticeable fraction of guys with no crossovers, or 1 or 2 or 3 or 4, even in some cases. And so yeah, there's a whole lot of different complications you could add into the simple model. As we discussed, you could have within a single clone of a cell line, you could have you could model different subclones with different de novo mutations. Yeah, so there's a whole lot of different variables you could tweak and add on, which you may very well want to do if you're going to apply it in real life. You'd also want to, I don't know how you would do this, but you want to make the thing robust, because when you crash this into real life, a lot of things will probably go not as you expected. And so that's partly modeled by the randomness that you put into the model. I mean, for example, you could make the whole thing more complex and say, well, sometimes cell lines just die, or sometimes you are mistaken about the sequencing. The sequencing is mistaken, or etc. Yeah, so it's a big, complicated thing to simulate, but I think it's mathematically interesting and computer sciencely interesting and biologically interesting, philosophically interesting, artistically interesting, politically interesting. So, yeah, pretty good. Well, thanks again to Rareș for that conversation, and see you guys on the flippity flop.